Welcome to the Conversational Flow, a KCAS Bio podcast.
Hello, and welcome to the 24th episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian, and I've been in the flow cytometry space for over 14 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe and partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri, so we're truly all over the map. We're amped up to have you listening to the 24th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off for episode 24, Brian is going to go over some news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got an esoteric list of articles to discuss today.
What makes them esoteric?
Well, they're not all cell therapy, so that's all. That's always a big thing for me. But this is another one, Adam, where you selected some of the articles. So
I feel like I'm going to be judged again.
No judgment, but that's fine. I call it esoteric. And I'll have to look up a source to figure out exactly what that means sometimes. But
well, here, let's hope then, at least this is my hope, that since I'm doing the agenda again this time, that there is no impact on your health or the general labor market, stock market, as it were, last time. Because obviously my agendas have that sort of power.
It's true. Gosh, are we expecting any other large stock market shifts? I don't think so. I think we're set. So with that, we will jump right in here to this latest medical research news. And the first item that you've got is the Aspire Biopharma breakthrough for their sublingual aspirin treatment.
Yeah, so this was interesting because it's essentially what they call a sublingual aspirin treatment to delay or prevent heart attack. As we know, there have been recommendations for taking baby aspirin once a day for reducing the risk of a heart attack. I had seen some further information that there's more nuance than everybody should take it at a certain age, right? The certain risk and bleeding risk, things of that nature that weigh into it. But specifically here, what this is for those that would take something like this, it actually works faster than just your standard baby aspirin. I think under the treatment and stuff was about 2 minutes before showing some modification here, essentially TXB2 inhibition, which essentially translates into better clinical outcomes. And so the ability here, since it is faster, is if you're having an issue, you're able to take something like this and perhaps it's able to better prevent negative outcomes, right? So the longer you're going through and having a heart attack, the more damage that could be done. And so this could reduce some of that damage. Now, they are still at the point where they're going to go through further testing. And so it would be interesting to see how this plays out.
And emergency medicine is always a little bit fascinating to me because we so often work with oncology and especially in clinical trials. We can be very careful and selective about what type of concomitant medications we have to worry about. But with emergency medicine, you get people with an acute myocardial infarction, i.e., a heart attack. You can't stop and understand treatment history or screen them for other medications or screen them for other indications. You just got to go and do what works best and fastest. And this definitely fits within that regime, right? In terms of working within 2 minutes here.
And two, this would be some, like, to your point, it's emergency. Whereas that whole paradigm of taking something every day as a prophylactic, it's much different. Like, the calculus is different for somebody here who's actually having a heart attack. They would then need some sort of treatment, and so there you would want to have the platelets stop aggregating, or at least aggregating to a lesser degree, so that it doesn't compound that problem.
Sure. I wonder how much of a consideration it is on whether or not this leaves the system quickly as well. That way you can sort of go on to next steps once the patient is somewhat stabilized.
Yeah, that's a good question. I imagine that since it is like aspirin, that it is something that the aspirin you have to take every day to continue that effect. And so I would fully expect that this would have some sort of half-life very similar there.
Sure. Yeah, but in terms of something that you could have on hand that could be shelf stable, that you could then give to someone that wouldn't be able to potentially swallow a pill or do anything else, and still have it take effect, it is definitely a meaningful treatment. And heart disease is still one of the biggest killers, especially in the U.S. and other developed economies that we need to take care of. So glad that they're finding some more ways to address it.
Yeah, and something a little bit off the beaten path for us, right? To your point at the outset, we've done so many chimeric antigen receptor stories, and it doesn't mean that that's the only important thing in pharmaceutical research. And so, kind of why I wanted to gravitate. You'll see a bunch of things here, a little bit more off the beaten path for us.
See, esoteric.
Mm-hmm.
But that being said, for our second item, we're going straight back, not to the cell therapy path, but at least to the oncology path, right? In terms of this algorithm that's reconstructing evolution and clinical progression of tumors for cancer's black box.
Yeah, it's looking at methylation marks, and so you can. One of the things I've always been interested in is evolution, right? And how the interrelationship of various populations and specifically human evolution. And you look at genetic changes over time, and that gives you an understanding of drift and, connectedness of populations. And here, what we're looking at is methylation marks. So when DNA is methylated, it specifically can cause changes to the nucleotides, essentially by breaking it down, and so causing it to go from one base to another. And so they can track, or what they're looking at here, I guess, is they're tracking those sorts of changes and being able to go back to the originating cell. What this does is gives an ability to then look at the projected progression here, really understand what risks there might be, and could also give a way for diagnostics, right? Looking at, okay, well, these are the sorts of traits, the methylation marks that indicate this sort of tumor or these sorts of issues, and then, kind of come up with a risk profile.
It's interesting, right? Because we of course do a lot of work on things like BRCA and some of the other genetic markers and risk factors for cancers, but those are pretty gross changes, right? Or significant changes where there's whole base pairs missing, or there are specific mutations that are seen.
Mm-hmm.
Whereas this is something that's what we, I guess, previously thought of as more of a transient modification, where it's just basically turning the on-off switch, if you will, for how strongly that gene is expressed. So it makes sense that would be a much more common change, because it is just that much less severe for the cell. So I'd expect it to be happening more, and it makes sense. I guess I'm always biased against methylation, though, because in grad school I did quite a bit of PCR testing and things like that in some of my cell lines and things. And every once in a while I think about methylation, but could not get it to work for the life of me. So I guess I'm just bad at methylation testing, and I'll leave it up to the experts to do that
Well,
better, right? But
it's interesting that you bring that up because I was doing more reading into this. I was much more involved with HDAC, histone deacetylases, as it related to protein expression. For example, we use an HDAC inhibitor to increase transient protein expression, and so here I think they're actually going just beyond methylation itself to the methylation mark, what it does to the DNA sequence. And so it can actually change the base. So it's not just the on-off methylation. It goes a step further there.
gotcha. Okay. Yeah, exciting stuff, and anything that can get us away from single cell sequencing. As cool as the technology is, it is definitely right now relatively time consuming and expensive. Right, where again you can frequently see it done in the context of large clinical trials where you've got specific translational questions that you want to answer, and then it's a great way to get really in depth answers to those questions that way. But
Mm-hmm.
from a clinical practice perspective, something that can be done in bulk is a lot more robust and promising here. So
Yeah. I'm curious, what was the target of your methylation studies? What were you looking to do?
I can't even tell you. All I know is that I had very good experiences with PCR and RT-PCR and very bad experiences with methylation testing. So
It's the reason I asked was just knowing that at least with the acetylation, that what we were targeting, we're doing protein expression. It was very much strictly an on-off sort of thing. But in reading through methylation, there's more ambiguity to it. The region of the methylation, whether the presence of the methyl group actually increases or decreases transcription. So I'm not surprised just when you say that there was some difficulty there. It seems much more of a vague topic than I had anticipated.
sure. I mean, definitely a little bit more nuance there and a little bit more of an advanced technique. If we ever want to talk about it in depth, I say we bring on Carrie from our Olathe team, and she can bring her expertise and experience with not only PCR and RT-PCR, but now ddPCR and things like that too, right? So,
That sounds fun. Yeah.
yeah. Um, all right, and then sort of moving on to investments. The first one that you've got is Novartis buying a U.S. biotech in $1 billion deal range.
Yeah, so I'm going to skirt around the name of the key asset. So the company is Tourmaline Bio, and like you said, 1.4 billion, offering about $48 a share here. The key asset is Passy Beketug. Oh, no, I killed that one. It's a monoclonal antibody, which I got to say, so many of the monoclonal antibody names are just so difficult to pronounce. And I try to pride myself in being able to pronounce things pretty well, but this one was a bit of a challenge. An anti-IL-6, IL-6 is interleukin, generally known for creating fevers, right? It's one of the cytokines that is known for cytokine storm when people are given chimeric antigen receptor therapy. And so here we're doing is an anti-IL-6 that developed for atherosclerotic cardiovascular disease. All right. And so has shown some strong phase 2 trial data so far and has a pretty high reduction rates, 85, 86% for the C-reactive protein, so high sensitivity C-reactive protein at 90 days compared to placebo. And really looking to expand here into inflammation and the reduction of inflammation in this space. That is one of the areas that's somewhat of an unmet need.
Sure, and IL-6 being one of those primary cytokines related to inflammatory responses and things like this, where any presence of that usually indicates, a little bit of an immune cell activation. And this space, as we've talked about a couple of times, inflammation falls right into the autoimmune space, and is just a huge area of investigation where if you can show control of inflammation, then that's sort of treating the symptoms of some of the autoimmune diseases, and or helpful for, as we discussed with aspirin and others, cardiovascular risk as well. So trying to target some of these other long-term chronic types of indications.
All right, and as you were saying that, I was going over in my head how to say this. So, Passy-Beketug. I think that's how you do it. So,
I I agree. I'm not going to repeat that to you because I trust wholeheartedly in your interpretation. But yeah, I think that's right. And this one I'll stack up there again with the aspirin because one of the things that's unique or really advantageous here is the dosage format.
mhm.
And we talked about this a couple times where compliance is a huge factor, and especially on a drug that you might need to take for an extended period of time. Um, if you make it difficult for patients or you just don't make it easy for patients, then the chance of this drug being taken appropriately goes down.
Mhm.
And if it's not taken appropriately, the chance of it being effective goes down as well.
It's interesting though that you even brought up the aspirin part. So, in looking up the information for this podcast, one of the things that I found was a study that was comparing whether or not the baby dosage was at 81 milligrams versus the full adult dosage of aspirin, over 300. Which one is better? And in the one study, they had mentioned specifically that the baby aspirin didn't seem to have any reduced impact compared to the adult for the prevention of heart attacks, that it seemed just as effective. The problem was that so many people on the adult dose would actually change their dosage. They wouldn't adhere to the full dosage. So, as you know, I mean, sometimes you could have gastro issues with aspirin, reflux or whatever, stuff like that. And so the baby aspirin, the lower dose is generally more tolerable there. And so imagine they didn't specifically dive into too much of that detail, but imagine that might be a driver. But I thought it was interesting to your point that even when it's just a difference in dose, you're still taking the pill. It's just a matter of what that impact might be. There might be some discomfort or whatnot that makes it so they don't adhere.
Yeah. I mean, just like with exercise, the most effective exercise is the one that you can do repeatedly, right? So it's a mixture of convenience, enjoyment, all sorts of other things. And here with taking a drug, again, that, I'll call it convenience, but of course it's more than that, right? Like it can be severely disruptive in your day, depending on how you need to take these. But if you can improve patients' access and compliance, then you can usually get a much better result for your drug. So I'm excited to see antibodies, making sure to take that into account. And it's a good tie-in to potentially cell therapy because, of course, there you can dose once or small numbers of times and get really long-lasting effects. So
It's
it's back to
interesting.
our meat and potatoes there.
I like that you brought that up because it was a partial topic that I was thinking of with our next news article, which dives into gene editing. And, you know, being in the pharmaceutical industry, you can't help but hear at times that therapies are withheld because, if you cure cancer, then the pharmaceutical companies aren't going to make any money. And here we're talking about chimeric antigen receptor, where the goal is to get durable, lasting treatment. So you get that injection the one time. Now, granted, I mean, they're fairly expensive at this point, but there's a lot of money being invested in trying to make them simpler, to make them more accessible, make it cheaper. And it's the same thing with gene editing, right? You're changing the DNA sequence so that the patient is cured. And my nephew, having gone through this and seen the impact, where prior to the gene therapy he was bedridden as an infant, he couldn't even move. Like, when people think of an infant, they're sleeping all the time, it wasn't that. Like, he couldn't eat, he barely breathe. I mean, just he could only lay there. And now with the therapy, I mean, he had been able to do T-ball and things of that nature. It's just an incredible increase in his quality of life, and surely the increase in quality of life of his parents, my sister and my brother-in-law. And so that's why I say that these are evidence of these therapies that really could be completely life-altering, that don't necessarily rely on constant treatment, constant dosing.
That's it, right? And again, indications that had just absolutely no way to address otherwise. So bringing in what we would generally term undruggable targets and bring those into the realm of things that can be treated with modern medicine. So, wild to see this going. So again, this is around the Lilly deal to buy the gene editing company Verve, and their technology works with CRISPR. So they're delivering CRISPR and usually a guide RNA sequence.
Yeah, and there are several genes they're looking at. Verve 102 specifically is anti-PCSK9. It's a phase 1B trial right now. And so looking specifically here at their cardiovascular profile and trying to reduce issues there specifically. Let's see.
Now, they are using LNPs to deliver their genetic payloads as well, to guide how the CRISPR edits in cells.
Uh-huh. Okay.
So that's as opposed to any type of viral delivery system or other things as well.
Right. And as you know, the various delivery systems have their pluses and minuses. The viral systems, for at least many of the earlier ones, you couldn't do repeated dosing just because the body would then reject the therapy. And so that's why investigations such as this is really important for identifying that.
And again, bringing it back to cell therapy, of course, Lilly did previously buy ESO Biotech, which had a LNP delivery system to create CAR Ts, right? So
Hmm.
this sort of keeps going along that same trend of if we're going to edit anything inside the body, then apparently Lilly is leaning a little bit more towards the LNP route as opposed to the viral delivery route.
Hmm. Now I think our audience has finally realized why we do so much chimeric antigen receptor stuff. I went off the beaten path on stuff. Not one of them actually mentioned chimeric antigen receptor, and every single one, I think, or just about every single one you've mentioned, cars. And so you're very much a car person.
That's it. All about cars, high speed cars, in vivo cars, car and Ks, all of them. So
Don't get me wrong, I love them too. I just think it was funny. When I put together this agenda, you had a look on your face like you were almost sad that there were no chimeric antigen receptors, and so you made it happen. And I'm glad to hear you made it work. So,
indeed. Anything else that you wanted to bring up about CRISPR gene editing and Verve and Lilly's sort of strategy within the gene editing space?
at this time, I think we've covered most of the topics there, and it just, I think from a market perspective, it is interesting to see how the larger companies continually acquire assets, acquire companies, making sure that there's funding behind these assets to be able to get them through clinical stage. But you see it in both ways. And I think we've even talked about this, where they're acquiring assets, but then also the divesting assets at the same time, constantly monitoring the pipeline and making sure that things are being able to be supported, that to their minds, a lot of times have the highest probability of success, right?
Yep. Yeah, and as you said, gene therapy is definitely fallen on some difficult times. I'd say probably even more so than the rest of the biotech market. So good to see that some of the larger players are still investing in the technology and buying what they see as good value companies as part of that. So hopefully
Right.
that will keep it going and keep more of these sort of one shot full treatments going as well.
Mm-hmm.
All right. Excellent. And I guess next up is KCAS news. And big things, of course, from the Philadelphia site for KCAS is we're definitely going to ICCS, so the International Clinical Cytometry Society, as well as SITC, so the Society for Immunotherapy and Cancer. Is that right?
Believe so.
Ah,
I wasn't
gosh.
expecting you to ask me that about that.
No, yeah, Society for Immunotherapy of Cancer. Okay, sorry, I couldn't figure out how the C fit into things, but there we are.
It's funny because I'm thinking about like the next thing that we're going to talk about. And you asked me a question. I'm like, oh, let me wait. What was that again? Yeah. So ICCS. This is nice. Last year we were in Seattle for that. Connected with many colleagues, and I think it's very informative. Very much focused on the clinical space. Obviously, with us being in CRO, we support clinical. This is tangential some of the stuff that's in there, but it helps to understand best practices and drive the science. I know many of our colleagues in pharma, academia, as well as CRO space are there. And so we're lucky this year it's in Philadelphia. And so we'll have a few of our team going down there and meeting up. Always looking forward to that.
That's it. And our team will be presenting some posters about cell sorting for AML, how to interpret RO assays, and I believe also on some of our pre-validated panels that we rolled out globally as well. So definitely some exciting talks that will be given, and we'll be excited to bring back all of the new information from those conferences as well. And since it's in Philadelphia, we're looking forward to hosting. So we'll definitely have some good dinners and meetings with all the people that'll be at the conference there.
Anybody from MAFCA?
Oh, yeah. So MAFCA is the Mid-Atlantic Flow Cytometry Association, and we are centered really around the Philadelphia area for the most part. So I would say probably a slight majority of our members will be there or in nearby meetings at some point during the conference.
Excellent. Yeah, it's always good to catch up. And I'm be honest, I knew that was the case, but wanted to give you the opportunity to give a little bit of a plug there.
Always appreciate it. All right. So, other and related, I guess, to some of our posters at ICCS is this paper on accessible high-speed image-activated cell sorting.
Yeah, so this is a little bit different for us, right? We don't have any machines like this. We're obviously interested in it. So this paper's Trends in Cell Biology. The impact factor as of last year was 13.06. So one of the higher ones that we've done, Tara Kuhn et al. And so looking at getting images of the cells as they're going through and then using that as a means to sort. Now, as you can imagine, and they do a fantastic job of outlining the struggles here, is obviously this has got to be data heavy. You're talking about so many different images. You're just trying to take image after image of cells, but they're flying through the machine. You're talking about upwards of 15,000 events, and trying to capture that image and then make a decision on the image as to whether or not it's something to be sorted. And if you're doing multi-way sorting, where does it get sorted? And so there are multiple ways that they outline that these images get captured. Now, they did reference something that we talked about before, ghost cytometry, which I frankly think is one of the coolest names out there. They mention that just because they don't ghost cytometry doesn't generate a 2D image, right? It infers it through machine learning that then makes a decision. It's I got to say, it's pretty complicated. It's really cool with the technology. It's just the fact that you don't even make an image, but you rely on fragments of an image to be able to make an informed decision and then utilize that. Where since it doesn't make the full image, it wasn't captured as part of this paper, right? What they're looking at are systems that can look at that the full image. And so why of our overriding topic here is sorting, and so that's kind of tied into this paper. I like this paper just because it's something off the beaten path. It's something that we really haven't gotten into, but then it also opens up the door for us to talk about cell sorting. And a lot of people know FACS, flow assisted cell sorting, and so here we can talk about kind of that triangle of sorting. You've got three things that you can get out of sorting. You could get purity, you could get yield, and you can get oh my goodness, where did
Speed.
Speed. Sorry, yeah, it was I just was looking at that wrong. And so you can get 2 out of 3 of any of those points, right? You can get high purity and high yield, but it's going to be slow. You could get high speed and high yield, but the purity is going to be slow. And so it's just going through those and understanding what you're going to get out of each one of those, what you're looking for. So for example, if you're going for genomics, you really want to have a pure sort. Whereas if you're looking for cell culture, obviously you want to look at speed, or somewhat pure. It depends here in cell culture. Like if you're going to do single cell per well versus bulk sorting, those are some considerations there. But and then finally, if you're looking at proteomics, purity and cell recovery are both really important. Clinical diagnostic, you're looking at purity, sterility. So all those sorts of considerations. Anyway, I want to stop there for a second. I have a whole bunch more on sorting. Brian, wanted to let you jump in either on the paper or sorting.
Yeah, I think if we look at the paper, as you say, the wild thing is when you think about how quickly this is happening, you're looking at thousands of events per second. And maybe I'll talk about conventional sorting and then go into image-based sorting. So thousands of events per second. They're going past a laser and detector. That detector is analyzing the signal and triggering some type of downstream action to move the cell into one of several sort of downstream collection bins, right? So this has to be extremely fast, milliseconds or microseconds, even in terms of how quickly that decision needs to be made and acted on. So that really limits what type of criteria you can set for how that signal should be interpreted. So it needs to be, X amount of the PE channel and Y amount of the FITSI channel, if you will. And that's how it has been always in the past, where our sorting rate has been sometimes limited by the data acquisition rate and that sort of machine decision making and actuation rate. Here, that gets more complicated because instead of just that binary, how large is this voltage signal, compare the two and go, that we've been able to do with circuits forever. Here, you're looking not only at that, but then generating an image, analyzing a portion of that image, and making that decision, again, within the time frame that the cell passes from the detector array until it gets to the sorting mechanism.
And the thing I would liken it to is anybody who's counted on a hemocytometer, right? You're looking in a microscope at a bunch of cells and you're counting the live cells and you're counting the dead cells. And then you have to do the 4 different fields and then, figure out how many cells per mil you have, do all the calculation. And how long just doing that one thing, and what kind of data are you getting? You're just looking at the cell, but you're not really making much more of a qualitative assessment than that one allowed trypan to go in the cell, so it's dead, versus that one is a nice shiny clear cell, so that one's live. This is even more information. And then one of the things they talk about on top of that was some of the options are to have fluorescence. Everything so far looks to be focused on the 488 nanometer laser. And so anything that's excited there would then be part of that. There aren't, any of the other lasers at this point in time. But certainly is something that would be worked on, I imagine. And so trying to take that image that you would see from a microscope and quickly just look at that one cell, like you said, passing by the optic and interrogating it for various features, if it's that fluorescence, so on and so forth. And so, yeah, there's various versions out there of how this gets done.
That's it. And I can hardly imagine even opening an image file, let alone opening an image file and isolating parameters of interest and then sending that signal back on like yes or no within the time, again, that the cell passes between the detector array and the sorting array. So just wildly fast image processing and circuitry there. So kudos to the vendors.
And do mention about there being a limitation regarding cell size, and that's something that has to be factored in because of the fact that obviously there are a wide variety of cell sizes out there, and so just another consideration. But I'd certainly see this being an add-on, like another thing as it evolves. Being very useful, right? I think in the past we've talked about imaging cytometry being a nice to have where it gives you that yes or no answer. Okay, what we saw in the readout on this 2D plot matches up what we see on this image. We really do see green or whatever. And so I thought that's all interesting. But here, some of the other things that they talk about is being able to interrogate cell phase. You can do that just flow cytometry as well. But getting the actual images, I think, takes it to another level, gives it a bit more granularity than you would otherwise have.
Absolutely. Maybe I'll call it two because I don't know if I fully go for the cell cycle analysis. Maybe it is, but the two killer apps that I see are subcellular localization, so understanding if a marker is on the cytoplasm or the cell surface or the inside the nucleus of the cell. With this type of technique, I don't know if you've got the resolution to go deeper than that, but it's certainly can do that sort of large scale localization.
Mm-hmm.
And then the second killer application is cell to cell signaling, where here you can look at doublets and see a lot more around the interface of that cell to cell interaction to understand what's happening there.
Mm-hmm.
So those are the two real killer apps that I see for this type of technology. There's just not that many research questions out there that are targeting that are intended for flow cytometry. Because I think also high-speed microscopy has come up relatively quickly as well. But I can definitely see now that this technology is available, people can start asking more questions and understanding if those answers are important or not compared to a traditional cytometry approach.
Mm-hmm. Yeah. And then just going back to cell sorting, I think it's something that I just wanted to talk about as well because we obviously have that as an offering, right? Here, a shameless plug again as far as business is concerned, but we do that for a lot of downstream analysis, sending
A
away
lot of
for.
downstream analysis of what type of therapy there, Adam?
I'll let you say that because I think you're much
Cell
more.
therapies, Adam. Cell therapies, CAR Ts, all sorts of different things. So
And so right there, RNA sequencing, right? But as I mentioned too, we can do enrichment of populations. We can do single cell per well sorting, so that you're for clonal generation of cell lines. There's just many applications, and constantly getting varied uses for it. I think. It's one of those things where I started out in flow cytometry because of sorting. When I was previously at J&J, I was generating stable cell lines and initially was doing it by Western blot and really was just very laborious, time intensive, and the amount of information that I was getting wasn't nearly as much as flow. And a couple of people there, a couple of my colleagues had turned me on to flow cytometry and were helping me get up to speed there. Not only they were running it at first, obviously, as a flow cytometrist, you don't really want somebody who doesn't know what they're doing touching your machine. And I was thankful for that. But then I obviously learned how to do it, and then learned sorting, and what really helped me to generate better tools, right? Because a lot of times that's what you're doing when you're generating stable cell lines. You're transducing them with a target. You're making a tool. And so a lot of times you could fine tune using sorting, where you could get high, medium, and low expression, or clonal, things of that nature. And so mainly a lot of the considerations there when you're talking about sorting, the type of cells that you're sorting, specifically whether or not you use a smaller or larger nozzle. We've already talked about whether or not you're going to use purity versus yield, and then a lot of the
Well,
considerations.
and with the nozzle, if we can maybe dial into that for a second, you use what a smaller nozzle for smaller cells, as you might imagine,
Mhm.
and
Also higher pressure, faster sort.
sure, yeah, no, if you want to get a lot of cells through very quickly, then that is usually the way that you would go. Um, I don't think you would want to use it for like extracellular vesicles or other things like that, but I don't see why you couldn't, as long as you got maybe a smaller nozzle even than normal. And then for larger cells, so anything over about 20 microns, then you'd want a very big nozzle, so something like a 100 micron nozzle.
Right.
So if we're talking about this imaging based cell sorting and looking at multiple cell to cell interactions, I suppose you'd need to use a relatively large or maybe even specialized nozzle to avoid disrupting that signaling.
Yeah, as you just mentioned, above 20 microns, we're talking about a 100 micron nozzle. So there is some room for doublets, but anything beyond that, obviously, you're going to start to get to the point of clumping. And anybody who has done sorting knows that a lot of times, if you're on the machine for a long time, the cells can easily just start settling out. And so, you might pause it, you might mix it, things of that nature. But I mean, your point is definitely valid there.
Yeah.
And so the next thing I was going to talk about were the panel design. And again, the same considerations you have for normal flow. Rarer populations, you want to have your brighter floors. One of the considerations, if there's autofluorescence, you want to go far red to avoid overlap, and just really carefully designing your panels.
Yeah, and with cell sorting applications, definitely surface stains are by far the most common. And also, generally, will run unfixed, right? Because most of the time, you'll need to do something with the cells afterwards.
Mhm. Yeah, and I mean that also then comes into how do you collect them? So if you're going to be doing some sort of culturing, you'll collect them in serum coated tubes. One of the things that I had done in the past was also collecting them into anti-anti, so antibiotic, antimycotic, and letting them culture in there for a couple of weeks just to be safe. Obviously, we go through a lot of cleaning procedures specifically to prevent any contamination there. But it was just, for me, it was always an extra peace of mind because you go through so much effort of generating your cultures and then sorting them that you hate to lose them for that random issue, for whatever reason.
Well, especially if you're doing like a single cell sort, right? Because then if there's even a single other bacteria, it can easily outcompete your stressed out single human cell that you put in there.
Yeah, exactly. And then if we got downstream omics, you've got RNase free buffers. So making sure that you targeting whatever you have downstream, you're using the proper reagents there. So another thing that comes up is post sort purity checks. That usually is more so if you have enough sample after the fact to be able to do that. A lot of times we may get sample that's just really precious, and clients don't want to do a purity check just because of that. It's not an absolute must-have. As long as you've set up your machine, you can be confident about the gating appropriately. You definitely do test at the beginning on controls or something like that. And so just another thing to consider.
I wonder with image-based cell sorting if those post-sort purity checks work the same way, right? Like I think to myself, are there enough unique characteristics that you could say, hey, that's cell Greg, right? And you see him one time, and then you do your purity check, and you're like, hey, that's Greg again. I imagine not, but
well, I think you have something similar with index sorting.
Sure.
And so you can look on a plot and point to that dot right there and see which well, for example, if you did a 96 well sort, where that went to. And so, but as humans, obviously like visual confirmation. And so if you, to your point, you're getting those images and you're seeing them as they go through, like, all right, I know it was definitely that sort. Or that cell. It was what, Greg, was it?
Greg the cell.
Yes.
Okay. Interesting. And I guess on one of our next episodes, we'll have to cover barcoding as well, right? Because that's the really the easiest way to find Greg, and really be very specific about it, right? Because you can get a combination of dyes that can be sort of uniquely associated here. Yeah, but no, cell sorting is fascinating. From a hardware perspective, it is certainly the highest tech thing where you need everything to be going exactly right? Where you need the lasers aligned exactly, you need the detectors, ready to go and compensated. You need to have your gate set ahead of time to be able to make those decision criteria. And then the instrument, again, the data acquisition rates and the decision making rates of the instrument are just phenomenal in order to be able to do this crazy feat of looking at each one of these single cells and then figuring out where it should go and getting it there. Again, faster than any human could ever do on a microscope, and then with a little tweezer trying to pick it up and put it into the right bucket there.
Yeah, I don't think it would work that way. I just remember getting my first sorter was Fax Jazz, and that thing was intimidating, right? You got all the buttons and knobs all over it, especially too when I was really just getting into Flow. I had an Accuray C6 and was doing that, just a few colors after having been doing years of just analysis of data. And then going out to was the BD training in Milpitas and doing that and the advanced sorting training. It's amazing how you can get comfortable with it. But for my money, there's nothing better than learning on a Fax Jazz for sorting, because it is the least automated platform out there. And so everything that you're doing. You have to know what you're doing. You have to understand each component, because the computer isn't doing it for you, from aligning the lasers and the AccuDrop delay and all of that.
It is like the very manual version of a flow cytometer, right? It is stick shift, manual windows, everything is user controlled, and an engineer's cell sorter, if you will. Definitely not one of those Apple fans at that point.
I got to ask though, did you drive stick? Do you know how to drive stick?
I do not, Adam. I'm a millennial, and that died out in the U.S. at least well before my time. So
I'm Gen X, and I had my nephew teach me how to drive stick. The thing is, I don't have a car that's stick. My car is automatic, right? And so he taught me, and I haven't done it since then. So I don't know how much. All I remember was you got to push the clutch all the way down, right? If you just leave a little bit out, it just doesn't engage, and it was the most frustrating thing. He's like, "Dude, push harder." It's like, I am. But yeah, Micah, if you're listening, definitely appreciated that. Thank you.
And Todd, if you're listening, this is why we have retraining protocols where if somebody has not processed an assay in a long period of time, they must retrain on the assay before they go into the lab with it. So,
yeah.
Yeah, excellent. Well, always enjoy talking about cell sorting and some of these advancements in cell sorting. We're talking about image-based cell sorting now. I believe that there is also spectral-based cell sorting. So excited to talk about some new applications of that in the future as well.
Absolutely.
For our next episode, I know on our podcast agendas we've promised what happens if you go with the flow too much several times now, but I promise that in October we will absolutely talk about what happens if you go with the flow too much. We'll have a special guest from the Chicago University Flow Cytometry podcast on, David Leclerc. So I'm excited to welcome him on the episode. And excited to really talk about how quickly flow cytometry can go.
Yes, looking forward to that myself. And I mean, you've kind of kept this under wraps. You've just teased it even to me. So I'm sure some of that will be new to me next month. And you get my genuine surprised face there.
Excellent. Um, uh-oh. You know what we didn't
The
have here? Yeah.
banter.
The banter. Yeah. What is the banter topic, Adam?
So the thing that's on my mind, maybe it's just because I'm hungry. I'm curious, what is your favorite food? Like, what is your favorite dish? If you had one thing to pick, and I don't want to say like if you're on death row or anything, but just in general, let's say you've been away for a long time. What's the thing that you're going for?
You bring up a good point that I used to say, like, what happens if they get hit by a bus? And this is a little bit morbid and pessimistic. So I've tried to consciously switch to saying, what happens if they win the lottery instead?
Mm-hmm.
But it's not natural to me. So I gotta really focus on
Well,
it.
it's
So
like
appreciate
that.
that.
And when you say somebody asks how you're doing, you say you're living the dream. I noticed
Oh,
you've
yeah.
changed that one too.
It's hard. It's hard to go against those ingrained patterns, but you can do it if you try.
Yes, you can.
So yeah, if I won the lottery and I was going to go out and do a celebration meal, I guess I always like what palak paneer. So it's an Indian dish with like a spinach curry around like a house cheese.
Oh, that's cool. I just had Indian last night. I had a spicy tiki masala that we made, and before that, I tried a new sauce. I think it was a korum simmer sauce,
Yeah.
and put some chicken in that. Had it on some rice. Oh, that was so good. So my daughter and I have been trying out different sauces of that vein, and so yeah, I sounds good. The only thing we forgot was naan. I couldn't believe it. We finished up, and she was complaining to me that I didn't buy naan. So I got to remember. Do you have garlic naan? Do you have just plain naan? What do you get?
I usually go garlic naan.
Mhm.
I try to keep all those other people away that have not eaten garlic that day.
And then get like vegetable samosas, mango lassi. You got to do it up
The
right.
whole 9 yards. Yeah. No, I mean, if you're going the rice and sauce and non route, then absolutely you got to go for all the rest too.
Maybe
So
some
yeah,
cheese dosa or something.
yeah. Now I know we talked about your affinity for pizza, but what is your favorite dish?
It's a relative of pizza. That's stupid. That's stupid. That sound. Ever since I was a kid, I've always loved eggplant parmesan, and I just can't get enough. Now, the other day, my daughter had someone over, a friend over, and my friend and I had gotten some eggplant parm, and she came up and she goes, "Oh, you can have the eggplant parm." And I'm like, oh, so disappointed. And I look at her friend, and this is the first time that I'm meeting this friend. And he goes, yeah, I feel the same. I was like, the two of you got to get out of my face right now. I was like, the only saving grace here is you guys saved extra for me. So thank you. But no, all kidding aside, I don't mind that she doesn't like it because I could eat an entire tray of eggplant parm. It is just so good, and so I'm glad that that's one of the better dishes that the cafeteria here has. In Building One, got to give them a shout out. They do some great lunches, and that one is I make a point to get just about every time I can.
Nice. Excellent. Yeah, no, eggplant parm is wonderful. There's a restaurant near my house in Smyrna, Georgia, that is famous around the city because they've got, I forget if it's an eggplant parm or a chicken parm.
Hmm.
And pregnant women, when they want to actually have their baby, they go to the restaurant and eat the parm dish, whatever it is. And there's an old wives' tale that if you eat it, then you will immediately go into labor and have your baby then.
Is it only that restaurant?
It is that restaurant.
I was going to say, because I've heard of people eating spicy stuff and what have you to try to induce labor. So that's a selling point of that restaurant then?
That is a selling point.
Imagine you got a lot of pregnant women eating there.
It is strange when you go in. It is not normal. Yep. It's pretty good. I didn't have a baby after I ate there, but it's
we'd be talking at a totally different kind of restaurant there.
We would. So
I think that's a great way to end this podcast.
That's it. Yeah, natural ending right there. So thank you to all of our listeners. Looking forward to any questions or feedback you may have, and also looking forward to welcoming you to our next podcast on going into the flow and sometimes too much.
Yeah, and may we all have good luck in that none of these topic agenda items impact you in any health averse way, and may the market continue to go up just because I did make this agenda. And thank you all for listening and dealing with my craziness.
All right, thank you.